Alright, hey everyone. So for this unit, we covered a variety of topics. So all the topics that are going to be on this exam, for the most part, are going to be mutations and the 4 types of them, the central dogma, DNA replication, transcription, translation, RNA processing / the gene model, meiosis and mitosis, biochemical pathways, and genotype / phenotype ratios. Alright, so for this video, I'm just going to cover some of the major topics we covered in recitations for the past 4 weeks. And so the first topic being genotype notation. Alright, so here we have one gene, 2 alleles, and it's going to be on the same chromosome. What the... Alright, so when a problem says that these are on the same chromosomes, what they're referring to is the same homologous pair. So here we have 2 homologous pairs. This is, for example, chromosome 1. And remember, homologous pairs are essentially a set of chromosomes which contain the same structural features, the same genes, just they may differ at the alleles. So they may have different DNA sequences for each of the genes. For example, this may be a gene that codes for eye color. Let's say that this allele is from the mother and this codes for blue eyes. Let's say that this allele is from the father and this codes for brown eyes. So essentially these chromosomes, they're essentially the same thing. They code for the same things. They just have different alleles. All right? So if it said, for example, there's one gene with 2 alleles on the same chromosome, you can draw out one homologous pair, and you can write the alleles and have them be on the relative same side to each one another. Because remember, structurally, these chromosomes are similar. So we would just put down the alleles, and an easy way to kind of think of this is you have it on the homologous pair, then you rotate it 90 degrees. So now you have the chromosomes laying sideways or parallel, and then you just write down the alleles that are on each of the chromosomes. And then for the notation, what you would do is, there's 2 types. There's this notation right here in which you just have a line, and this represents a chromosome. You write the allele associated with that chromosome, another chromosome, the allele associated with this chromosome. However, for the exam, you're going to want to write it in shorthand, which all you have is just a dash in between them to signify that they're on the same chromosome. So this dash equals these 2 parallel lines. All right, now let's say that there's 2 genes and 2 alleles for each of the genes on the same chromosome. All you do is you'd simply add a gene onto the same homologous pair that we did from up here. Now you would just follow the same steps in which you would rotate it clockwise 90 degrees, and now you would have the chromosomes both parallel, and you would write the alleles associated with each of the chromosomes. It doesn't really matter how you write it. You can write A, B, like big A, big B, chromosome. And you can also do like little b, little a. It wouldn't, or I mean little a, little b. However you want to, darn it, little a, little b. That doesn't really matter. What does matter is that you don't get it crossed and, for example, write it down like big B, big A, little a, big B, because remember, these chromosomes are structurally similar. So what you're saying is that the gene for the B, at this gene you have an allele for an A gene, which wouldn't really make any sense. They have to be on the relative same side for each of the genes. The alleles have to be on the same side relatively. So the way you would write that down is you'd simply turn these chromosomes into lines, and then you'd write down the alleles associated with it. So you'd write, for example, big B, big A, little a, and then for shorthand, you'd simply just put a dash in between them and you write it the same way. Now let's say you had two alleles, two genes on different chromosomes. What this is saying is that there are two homologous pairs, and each of the genes are on this different homologous pair. So here we write down the homologous pair number 1, homologous pair number 2, and now we're going to write down the alleles for the A gene, and the alleles for the B gene. So now follow the same steps, just rotate it clockwise, 90 degrees, and then you would just write down this longhand notation, if that makes it easier for you to, I guess, conceptualize how it may look. And then when you want to write a shorthand, you have to write big A dash little a, then you have a semicolon. This semicolon signifies that they are on different homologous pairs, they are on different chromosomes. They are not on the same chromosome, these are genes which are located on different chromosomes. And then you would simply write down the B notation, shorthand. And I guess this is just a way for people to kind of conceptualize it and see it from a visual standpoint. It makes it a lot easier to understand it compared to just trying to figure it out and trying to think about it from a conceptual standpoint in your head. All right, going on to the second topic, which was DNA replication and the central dogma. Remember, the central dogma of biology is essentially that DNA flows in one direction, from DNA to RNA to protein. To get to DNA, you have to transcribe it, so transcription occurs. To get from RNA to protein, you have to translate it. So I guess that's kind of an easy way to remember it, like DNA and RNA. They're very similar in the sense that they use very similar nucleotides. So think about it as if like you're trying to write down a different language, as if you're trying to as if you're just trying to write it down. Think about RNA to protein, because when you're going from RNA to protein, you're adding in amino acids. So think about that as if you're trying to translate it into a different language, if that helps you remember it. So for DNA replication, you have a template strand and a coding strand. So a template strand essentially acts as a guide for DNA and RNA replication. So let's say, for example, we have a template strand right here. Let's say it goes 5', ATG, 3'. You have a template strand. Then you're going to have a coding strand, which is going to be the complementary strand to the template strand. Because remember, the template strand acts as a guide for DNA and RNA replication. So the coding strand, or the complementary strand, would be 3', TAC, 5'. And the coding strand determines the mRNA sequence. So the coding strand is essentially the same thing as the mRNA sequence. The only difference is you replace the T's with U's. So to get to mRNA, you would write down the same thing as the coding strand, just replacing the T's with U's. So it'd be U, 3', UAC, 5'. And here I have some extra examples in which the template strand is 5', ATT, CGA, ACC, 3'. The coding strand is going to be the complementary strand to that, so you just write down the complementary strand, which would be 3', TAA, GCT, TGG, 5', mRNA. Same thing as the coding strand, just replace the T's with U's. And something I want to note, when going from mRNA to protein, translation can only occur in the 5' to 3' direction. So if we look at this mRNA, it's going from the 3' to 5' direction. So in order to translate this, we have to flip it. So we would write down the 5' side, and then we would simply just write down, and we would simply just, I guess, read it backwards and write it down. So we would we would write down G, G, U. So as you can see, we did that here. UCG, we did that here. AAU, we did that here. And then we put down the 3' side. So this is how the mRNA would be translated. All right, going on to biochemical pathways. So a biochemical pathway is essentially a series of enzyme-mediated reactions that uses the product of one reaction to facilitate the next by acting as a substrate or a precursor. So what that means is you have, for example, in this example right here, we have molecule X. Enzyme A converts molecule X into molecule Y. Molecule Y is then converted by enzyme B into molecule Z. So you have a series of, so these right here are the enzymes which mediate the reactions. They facilitate the reaction from molecule X to molecule Y, from molecule Y to molecule Z. So in this case, the beginning molecule is known as the precursor. And the precursor is converted by enzyme A into an intermediate, which is molecule Y. Now this intermediate acts as the precursor for enzyme B. In which enzyme B converts this precursor for it, or this intermediate, into molecule Y, which would be the final product. Now, for example, let's say that we had a mutation that codes for enzyme A, and now it's non-functional, and we had the presence of molecule X. What would then happen? Nothing. There would be no growth occurring. And I guess it's important for me to mention that biochemical pathways, what you essentially do with it is you have a model organism, let's say it's yeast. What you would do is you'd mutagenize it, or you'd hit it with some sort of UV lights, or anything to damage the DNA sequence. Now, this organism normally would be able to grow on complete media, which is media containing the optimal growth necessities and nutrients. So it'll contain, for example, all the amino acids, all the nutrients, the main source of carbohydrates it would use, let's say it's glucose. It's the most optimal environment for growth. So normally this organism would grow on complete media, and it would also grow on minimal media, which is media containing the minimal amount of nutrients for growth. So it would be, for example, something like glucose and some inorganic salts. Normally, this organism would be able to grow on the both because it converts the glucose into all the other nutrients it needs through these enzymes, and then that would allow for growth. Let's say that after this DNA was damaged, it was only able to grow on complete media and not minimal media. That would mean that within this pathway, you had a mutation at any of the enzymes, perhaps one or perhaps two, and that does not allow for the conversion of one of the molecules to the other, which would then not allow it to get to the final product, which allows it for growth. All right, so let's say we had mutant enzyme A, and we had the addition of molecule Y. As we can see in this pathway, molecule Y comes after enzyme A, which means that if molecule Y was present and there was a mutation in enzyme A, that would have no impact on it. Enzyme A only converts molecule X to molecule Y. It has no effect on molecule Y going to molecule Z to the final product. Enzyme B does, which would mean that if you had a mutant enzyme A and molecule Y, growth could occur because enzyme A does not impact molecule Y and the conversion from it to molecule Z. Likewise, if we had a mutant enzyme A and we had molecule Z, then we would have growth at that path. Then we would have growth because enzyme A is right here, molecule Z is the final product, it's what allows for growth. If we had a mutant enzyme A, it comes after, if it comes a lot before it in the pathway, no effect, growth would occur. So for example, here's how some of the problems would be written out. You'd have the mutants written out in some sort of order, going this way, and then you would have all the medias on top. So the way to solve this problem is, first things first, you'd have to try and see you'd have to order it from least growth to most growth. So we can see at minimal media, this had the least amount of growth. We can see at tyrosine, this had the most amount of growth. So what this means is, since it had the most amount of growth, this must mean that it's the final product in the pathway. So for example, tyrosine in this case would act as molecule Z. It's the final product of these reactions, which would mean that growth would occur 100% of the time if you added this to the medium. We can see that HA had 2 spots of growth, MA had 1 spot of growth, 4-HPP had 3 spots of growth, meaning that HPP comes right before tyrosine, HA comes right before HPP, and MA comes right before HA, and then you have minimal media. So if we label it if we write it out from least growth to most growth, we have minimal media. The second most growth would be MA, third HA, fourth HPP, fifth would be tyrosine, and this has growth at every single mutation, meaning that it's the final product of the pathway. So going from minimal media to MA, we can see that at mutant gene TRYD, we had growth. So what this means is when we added MA to minimal media, growth occurred at this mutation. So what that means is this mutation, or this this enzyme is responsible for converting minimal media to MA. So since growth occurred here, we know that this gene is responsible for producing an enzyme that converts minimal media to MA. If we look at MA to HA, we can see that there was growth at TRYA, which means that the enzyme responsible for converting MA to HA is TRYA. For when you're going from HA to 4-HPP, we can see that TRYC had growth, which means that TRYC is the enzyme responsible for converting HA to 4-HPP. And going from 4-HPP to tyrosine, we can see that at 2 different areas, TRYB and TRYE, there was growth, meaning that both of these enzymes are responsible for converting 4-HPP to tyrosine. So you have 2 different enzymes that can convert 4-HPP to tyrosine. And this had growth at everything, meaning that it is the final product. Going on to Mendelian genetics. Mendelian genetics, this is essentially a way for you to organize phenotype and genotype ratios. And you're going to be told a term quite frequently. It's going to be called true breeding. So what that means is that's an organism that always passes down a certain phenotypic trait to its offspring. So for example, it's going to be homozygous, either dominant or recessive. Let's say we had a short individual, and his genotype is little a. If crossed with another short and true breeding individual, they're always going to produce an offspring that's going to have the same phenotypic expression. All of its offspring are going to be short. Likewise, if it was tall, it'd be the same difference. All right. So here we have an organism that's homozygous dominant at the A gene, homozygous recessive at the B gene. We're going to cross that with an organism that's homozygous recessive at the A gene and homozygous dominant at the B gene. So when you cross an organism that's homozygous dominant and homozygous recessive at the same gene, you're always going to end up with a heterozygous individual, or all heterozygous individuals. So here we have the organism, and it's homozygous dominant. At one of the genes, it's homozygous dominant. We're going to cross it with an individual homozygous recessive at the same gene. So little a. So we can see that all of the offspring are homozygous. They have a big allele and a small allele. So all big A, little a. We can see that at the B gene, it's the same thing. So the offspring are all going to be heterozygous. Now let's say we crossed the F1. We crossed an organism from this generation, from the first generation. 2 organisms. Now we both know, or we all know, that they are heterozygous at each of the loci. So now we're going to cross the heterozygotes. We have the A allele, or the A gene, and we're going to do a cross for both of these genotypes at the A allele, or A locus. So we're going to have a big A and a little a. We're going to cross that with a big A and a little a, and we're going to end up with 3 different genotype possibilities. This is going to be homozygous dominant, heterozygous, and this is going to be homozygous recessive. Now, for these organisms, for these individuals, they are always going to be, okay, so for these alleles, big A counts for red. If an individual or an organism has a big A allele, he's going to be red. A little a allele, he's going to be white. A big B allele, they're going to have long wings. Little b allele, they're going to have short wings. So these organisms with this genotype, they are going to be red. And we can see that the ratio for them, for homozygous dominant would be 1 out of 4. Individuals, heterozygous would be 2 out of 4. So what we'd do is we'd simply add them up. So 3 out of 4 of the individuals are going to end up being, or showing the red phenotype. 1 out of 4 are going to show the white phenotype. We can see the same thing for this side. 3 out of 4 of the individuals are going to have long wings. 1 out of 4 of the individuals are going to have short wings. So now, if you wanted to see all the genotype frequencies, the frequency for each genotype popping up, what you'd do is you'd simply write out the ratios for each of the genes or the different genotypes. 1 out of 4 of them are going to have big A. 2 out of 4 of them are going to be heterozygous. 1 out of 4 of them are going to be homozygous recessive, little a. And you multiply that with the ratios for the other genotypes. So if we wanted to see the ratio for having an organism with the big A, big B genotype, we would simply take the ratio for big A, multiply that with the ratio for big B, and we get 1 out of 16. So this would be the ratio of having an individual with the genotype big A, big B. We can do that for the rest of them, and we can figure out all the different genotype ratios and frequencies. And remember, frequency is just a decimal, so that would be 1 divided by 16, which I think is 0.0625. So this would be the frequency for this genotype. But then they would ask you, for example, how many individuals, or what's the phenotypic ratio of having an individual with red and long wings? We know that to be red, you have to have at least one big A allele. This right here signifies that the other allele doesn't matter. It could be either dominant or recessive. It won't have an impact because the organism is going to show the red phenotype. To have long wings, you have to have one big B because it's dominant. Likewise, the other allele doesn't matter. So we know that in order to be red, there's a 3 out of 4 possibility or ratio to have the red phenotype. So you take 3 out of 4 and you multiply that with the ratio of having long wings, which is also 3 out of 4. So that gives us 9 out of 16. To have the red phenotype with short wings, it is a 3 out of 4 ratio. Multiply that with the ratio for short wings, which is 1 out of 4. We get 3 out of 16. To have a white color with long wings, the ratio is 1 out of 4 times 3 out of 4, which is 3 out of 16. To have white coloring with short wings, the ratio would be 1 out of 4 times 1 out of 4, which is 1 out of 16. So the phenotypic ratio for this would be 9 to 3 to 3 to 1, because we can see that 9 individuals are going to have red with long wings, 3 individuals red with short wings, 3 individuals white with long wings, 1 individual white with short wings. And this is a typical Mendelian ratio. Alright, sorry guys, I had to cut this video because I got a bit thirsty, so I went to get a drink of water. Alright, going on to some of the practice problems. Alright, so let's refer to the Mendelian and Stahal experiment. What would we expect if DNA replicated conservatively, dispersively, or semi-conservatively? So, for DNA to replicate conservatively, what that essentially means is... Sorry, just give me a second. Alright, so for DNA to replicate conservatively, that would mean that the DNA double helix acts as a template for DNA replication. So the entire DNA helix. Let's write it down here. Okay, that's not good. So the entire DNA double helix acts as template for DNA replication. So what that means is if we were to have a DNA double helix right here, that's terrible. This is the best one I can do. This entire double helix, when it's replicated, it's going to act as a template for the other DNA double helix. So what we're going to end up with is two strands of DNA, one containing all the parental DNA, and the other strand containing none of the parental DNA, just being completely new synthesized DNA. The other part of the question talks about the dispersive molecule. Dispersive model. So the dispersive model essentially says that when DNA replicates into two molecules, it replicates so that both of the molecules are mixtures or hybrids of the parental and new daughter DNA. So here we have a DNA double helix. It is going to replicate. So we're going to end up with is a DNA molecule that's just, or two DNA molecules that look similar to this, in which it would just be kind of mixtures or hybrids of both parent and daughter DNA. And the other model would be the semi-conservative model. And this is the correct model. This model essentially states that each strand serves as a template for the new strand. So the two DNA molecules, double helixes that are produced from DNA replication, will contain one parental strand and one daughter strand of DNA. So here we have DNA double helix is going to replicate. So what we're going to end up with are two DNA double helixes that contain one parental strand of DNA and one daughter strand of DNA, the newly synthesized strand. This is what the semi-conservative model states. All right, now going on to the next question. Which of the two strands will have the higher melting temperature? So remember, the melting temperature is dependent by the number of hydrogen bonds that are present between each of the strands. So to determine which one has a higher melting point, we'd have to see which strand of DNA has more hydrogen bonds. The more hydrogen bonds, the more energy it takes to break those strands apart, the higher the melting temperature would be. So in strand number A, if we count up all the hydrogen bonds, we know that adenine and thymine have two hydrogen bonds holding them between them, and we know that guanine and cytosine have 3 hydrogen bonds between them. So this right here would have 2 hydrogen bonds, 4, 6, 9, 12, 14, 17, 20, 22, 24, 27, 29. So this strand A has 29 hydrogen bonds. For strand B, this has 3, 6, 8, 10, 12, 14, 17, 19, 21, 23, 25, 27. Strand B has 27 hydrogen bonds holding in place. Strand A has more hydrogen bonds, keeping it, stabilizing it, which means that this would have a higher melting temperature. Which of these strands is the leading strand template? A or B? And we know that the origin of replication starts right here. So which of them would be the leading strand? We know that DNA polymerase reads 3' to 5'. So here we have DNA polymerase. He's going to go in this direction. He's going to add on nucleotides 5' to 3', but it can get kind of tricky to kind of remember what way does DNA polymerase go, what happens. So just know that DNA polymerase goes from 3' to 5'. He follows this direction. So we can see that strand B is going 3' to 5', and the origin is right here, which means that DNA polymerase is going to go in this direction. So this strand, so strand B would be the leading strand. All right, here's the coding strand of DNA. This is incorrect. This needs to be a T. Well, okay, let's first convert this to mRNA. So we know that this is the coding strand, which means that it's essentially the same as the mRNA strand sequence. Only difference is that it has thymine instead of uracil. So to make this mRNA, all we simply need to do is just change the T's for U's. So we'd have A, U, G, UAC, CAT, CAU, AAG, GCG, 3'. So this would be the mRNA sequence. For this problem, I'm not going to do this portion of the problem. I don't have a codon chart with me. You can try that on your own. All right, going on to the 4 types of mutations. So there are 4 types of mutations. There are frame shift mutations, and this is either a deletion or an insert. You either delete or insert a nucleotide base. So for deletion, you would delete a nucleotide base. For insertion, you would insert a nucleotide base. And these are typically most harmful. The reason that they're most harmful is because they essentially shift the entire DNA sequence. So everything after that mutation would be shifted either one up or one down, which would then shift the entire amino acid sequence, which could produce a non-functional or a different protein. And then you have synonymous mutations, in which a mutation in one of the nucleotide sequences occurs and that does not change amino acid. So a nucleotide base is changed, but it still codes for the same amino acid. You have a non-synonymous, in which a nucleotide base is changed, and then that codes for a different amino acid. So change in the amino acid. And then we have nonsense mutation, in which when a nonsense mutation occurs, what happens is a random stop codon is added in the in a portion of the DNA or mRNA sequence. Alright, consider a standard gene model, what components end up in the pre-mRNA, mature mRNA, and what components are translated. So in the pre-mRNA, what we have is, we have the 5' untranslated region, exons plus introns, then we would have the 3' untranslated region. In the mature mRNA, what we end up with is the 5' untranslated region, the exons, and the 3' untranslated regions. So the introns are spliced out. The introns are insignificant. They do not code for the desired protein. In the mature mRNA, those are spliced out, and all we're left is with the exons. And the exons are what is translated. Now you have some other things that are added, like the 5' cap, which would be added right here. And you'd have the poly-A tail. I don't believe you guys need to know that for this exam. But in the case that you do need to know it, they essentially just add stability and facilitate the movement of this mature mRNA from the nucleus into a ribosome that's located in the cytoplasm. But for the most part, I believe this right here is all you need to know. Alright, the video cut out. Back on to the next question, which is how many chromosomes are in a cell at end of phase 1 of meiosis when 2n equals 6? How many chromatids are in the same cell at end of phase 2? When does the cell become haploid? Alright, so remember, meiosis, what you have is you have meiosis 1, meiosis 2. Meiosis 1, splitting of homologous pairs. In meiosis 2, you have the splitting of the sister chromatids. Alright, so here, let's say we have a replicated chromosome. This is considered one chromosome because it has one centromere. The centromere is the defining factor for what is considered a chromosome. So this is one chromosome with two sister chromatids. So if we were to draw the cell out currently in anaphase 2 or anaphase 1 of meiosis, we can see that we have a total of 6 chromosomes in the cell. So 2n equals 6, and 2n essentially means that it is diploid. So that means that there are sets of 2, and there's a total of 6 chromosomes. So if we were to find the haploid number, we'd simply just divide this over, so n would equal 3. So the haploid number would be 3 chromosomes. 2n just means that there's 2 sets, or that there's 2 chromosomes in a set, and there's a total of this many chromosomes. In this case, it's 6. So there would be 3 sets of 2 chromosomes, and we can see that we have 3 homologous pairs, 2 chromosomes per homologous pair. So in anaphase 1, there are 6 chromosomes. How many chromatids are in the same cell at anaphase 2? Remember, meiosis 1, splitting of the homologous pairs, meiosis 2, splitting of the sister chromatids. So here we have 2 of these cells. Let's say that they're in anaphase 2. They're about to be split. The sister chromatids are just the replicated portion of the chromosome. This right here is one sister chromatid. This has a total of 2. So we'd simply just need to write this out. Just need to count them up. You can see that there's 1, 2, 3, 4, 5, 6. So there's 6 sister chromatids in anaphase 2. When does a cell become a haploid? Remember, the diploid haploid number is based off of the amount of the set of the chromosomes. So since it's diploid, meaning that there's 2 chromosomes per set, it'd be 2n. To find the haploid number, you just simply need to divide over and you get n, which is the haploid number. So we can see that in this example, once the cell goes through anaphase 1, the homologous pairs split. So that is the point when they stop becoming diploid and they become haploid. Genotype and phenotypic ratios. In Drosophila, gray body color is dominant to ebony body color, while long wings are dominant to vestigial wings. Assume that the P1 individuals are homozygous. Work to find processes through the F2 generation. Determine the phenotypic ratios for each generation. All right, we're just going to do portion C of this problem. So assuming that the P1 individuals are homozygous. All right, so gray is dominant to ebony. So we're going to assume, since it's gray, it's going to be homozygous dominant. It has long wings, meaning that it's also going to be homozygous dominant for those wings. So big L, big L. It's crossed with another gray individual, homozygous dominant. This one has vestigial wings, homozygous recessive. So we're going to cross these. So we can see that at the G gene, the individuals are homozygous dominant. And they all have big alleles, meaning that all the F1 or all of their offspring at this gene are going to have the big alleles as well. We see that at the L gene, one is homozygous dominant, the other is homozygous recessive. If we were to write out Punnett square, we find that all the offspring are going to be heterozygous. So all the offspring are going to contain the genotype big G, big L, little L. Now let's cross this and find the F2. So we're going to cross the first generation with another individual from the first generation, so they're going to have the same genotypes. The G's still dominant, so we're still going to end up with big G, and so we're going to get 4 out of 4 with individuals. If we were to do a Punnett square, we're going to have big G. We have heterozygous individual. We have heterozygous alleles at the L gene. So if we were to do a cross for that, big L, little l, big L, little l, we would find that we're going to end up with 3 distinct genotypes. 1 out of 4 of them having the homozygous dominant genotype. 2 out of 4 of them having the heterozygous genotype. 1 out of 4 of them having the homozygous recessive phenotype or genotype. Now let's count out all the different phenotypic ratios, the different phenotypic possibilities. So we can only have gray color, so all the individuals have to be gray. We see right here that we have 2 genotypes that will produce long wings. So that's 3 out of 4 of them. If we add these together, that are going to produce long wings. We have 1 out of 4, which are going to produce short wings. So we can have gray with long wings, and we can have individuals that are gray with short wings. In order to get this ratio, all we need to do is multiply the ratio for having the gray phenotype, which is 4 out of 4, since all of them will be big G. Multiply that with the ratio for having long wings, which is 3 out of 4. What we get is we get 12 out of 16. The amount of individuals that can be gray is going to be 4 out of 4. Multiply that with the ratio of having short wings, which is 1 out of 4, which then equals 4 out of 16. So it would be a 12 to 4 ratio. Now, there's a common factor in this, so we can simplify it and make it 3 to 1. So this would be the phenotypic ratio for having gray and long wings to gray to short wings. If you wanted to also account for ebony long wings, ebony short wings, We know that none of the individuals are going to have those, so we could just put 0, 0. If we wanted all of the genotype, the different phenotypic possibilities. All right, when you cross it, and big A, little a, and big B, big C, little c, big A, big B, little c, what is the frequency of each of the genotypes? We're going to do one of them. Let's do A. So what you would need to do simply is cross all of the different genes for each of the individuals together. So for the A gene, all we're going to do is we're going to do a cross big A, little a, cross with the other genotype for the other individual, which is big A, big A. And the desired genotype is big A, big A. So we have 2 individuals, or 2 out of 4 of them, which are going to have big A, 2 out of 4. Then we're going to have big A, little a. For the B gene, we're going to do another cross with these individual genotypes. We have big B. Cross that with a big B and a little b. We're going to have 2 out of 4 ratio for having big B, 2 out of 4 ratio for having big B, little b. Then for C, we have, for the C gene, we have a big C, a little c for one of the individuals, and 2 little c's for the other individual. So 2 out of 4 of the individuals are going to have a big C and a little c, and 2 out of 4 of the individuals are going to have little c, little c. We want to see the frequency of having big A. So having big A, the ratio is 2 out of 4. So we multiply that with the ratio of having big B, which is also 2 out of 4. Multiply that with the ratio of having little c, which is also 2 out of 4. So if we just multiply this across, 2 times 2 is 4. 4 times 2 is 8. So it'd be 8 out of, I think, 64. It's asking for the frequency, so it wants it in a decimal. So what we need to do is we just simply do 8 divided by 64. So the frequency would then be 0.125. That's all you do for this. Going on to Hardy-Weinberg. So remember Hardy-Weinberg. What Hardy-Weinberg does is it tells you the genotype frequencies and the allele frequencies in a population that is at equilibrium, meaning that the population is not changing going from one generation to the other generation. All right, so there are two equations that you're supposed to use. There's a genotype equation, and there's also an allele equation. All right, so for the genotype for the genotype equation, that would be p squared plus 2pq plus q squared equals 1. And these equations are all in frequencies, meaning that they have to be decimals. They are percentages. All right, and for the allele frequency, it would be p plus q equals 1, and so p squared is the amount of homozygous dominant individuals, 2pq, heterozygous individuals, the frequency of them in the population, and q squared equals homozygous recessive individuals. For the allele equation, p equals the dominant allele, q equals the recessive allele. So for this question, it says that the occurrence of a rare autosomal recessive disorder is 1 in every 1,200 people. What is the frequency of the dominant allele? What percentage of the population is carried for the disease? All right, so what we're given is, we're given the amount of individuals that have this recessive disorder in the entire population. So one of the individuals has the genotype that is recessive. So this is giving us q squared. So q squared, remember, this is in frequency. So we know that 1 in 1,200 people have this disease. So Q squared would equal 1 over 1200, so the amount of recessive individuals over the total population, to give us the frequency of the individuals that carry the recessive genotype. I think that would be 0.0008, not mistaken. So Q squared equals 0.0008. Now, the question is asking, what is the frequency of the dominant allele? So we know that the allele equation is P plus Q equals 1, but we're given Q squared. So in order to get this to Q, all we simply have to do is square root it. Square root both sides, so now we get Q equals 0.03. So now we just use this and plug it back into the allele equation, so P plus 0.03 equals 1. So we get, if we subtract 0.03 on this side, on the other side, we get P equals 0.97. So 0.97 would be the frequency of the dominant allele. So now it asks what percentage of the population is carried for this disease. Now that we have both the allele frequencies, we know that P equals 0.97, Q equals 0.03. We can use these and plug them back into the genotype equation to get all the different frequencies for all the different genotypes. So now it says what percentage of the population is carried for this disease. Simply what we have to do is 2 times 0.97 times 0.03, and that would give us, I think, 0.05, which is 5% of the population is heterozygous for this allele. Now, if it asked you how many individuals are heterozygous in this population, you know the frequency of them, so 5% of the total population is heterozygous for these alleles. What you would do is you take 5% or 0.05 and you multiply it by the total population to get the amount of individuals that would be heterozygous.